It gives me great pleasure to speak at this virtual meeting today, and I'll present for you a brief summary of the genomic studies we and others have done on uveal melanoma, and how some of these findings may impact on choice of treatment, and particularly choice of immunotherapy for some patients. So most of you will be well aware, uveal melanoma development starts when one of these 4 oncogenes at the bottom left here are mutated in a melanocyte in the eye. These mutations promote cell growth, but are not sufficient on their own to lead to uveal melanoma. Additional genomic changes are required for that to happen. These initial oncogenic mutations lead to the formation of a nevus in the eye, and there are then 2 pathways shown on the bottom right here that the uveal melanomas can subsequently arise through. And these are based on the specific types of mutations and genomic changes that occur in those particular tumors. So these 2 pathways determine the possibility or probability of metastasis of those primary uveal melanomas. So there is a high risk and a low risk group of potential metastatic spread to other parts of the body. So the oncogene mutations can be thought of as drivers of the initial formation of a benign tumor in the eye, after which mutations in other genes such as BAP1, SF3B1, or EIF1AX, along with various chromosome copy number changes, for example, deletion of chromosome 3, or deletion of the long arm of chromosome 6, or gain in copy number of the long arm of chromosome 8, along with the associated gene expression changes that arise from these events, ultimately determine 4 very distinct molecular subgroups of uveal melanoma. And these were initially termed ABCD. Subsequently, a more refined analysis of a larger subset of tumors by the Cancer Genome Atlas Consortium refined these subgroups, and they termed them 1, 2, 3, 4, but essentially that's the same as the previous ABCD. So those subgroups really determine the likelihood of metastatic spread of the primary tumors. So if you look at the top left curve here, the light blue line in the survival curve, this is the survival for tumors that have no mutations in any of the prognosis genes and no copy number changes. And these tumors have no long-term adverse outcomes. In contrast, if you look at the orange line here at the bottom, these are the tumors that have lost chromosome 3 and carry BAP1 mutations. And these are the ones that have the worst prognosis. So the biggest question facing uveal melanoma patients and clinicians currently is how can knowledge of the genomics of uveal melanoma be exploited to determine or help develop new treatment options for patients. So for cutaneous melanomas, those on the skin, the last decade has shown remarkable improvement in treatment outcomes. And these have translated to increases in survival, some even called cures from cutaneous melanoma. Those treatments fall into 2 main classes: molecular targeted therapies and immunotherapies. And so for patients with advanced cutaneous melanoma, either combination targeted therapy or combination immunotherapies provide durable responses in about 50% of cases. So you heard earlier from Anthony Joshua that despite the large number of clinical trials of different agents for treating advanced uveal melanoma, the clinical response rates are disappointingly low. To a large degree, this is because many of the trials have focused on giving uveal melanoma patients the same therapies that are used for cutaneous melanoma patients. But these 2 histologically distinct subtypes of melanoma are very different. So it might be expected that the treatments for cutaneous melanoma will not work or be as effective in uveal melanoma. So the question remains, how can knowledge of the genomics of uveal melanoma be exploited to determine treatment options for patients. So to better inform on such possibilities, we sought to further characterize the genomic landscape of uveal melanoma by conducting a study in which we fully sequenced the genomes of 103 uveal melanomas and compared the sequences to the matching germline, that is the blood DNA, from the same patients. This busy slide here summarizes some of the key findings. Each column represents the data from a single tumor or patient. So the top panel, panel A, shows the tumors ordered from left to right based on the number of mutations in that tumor sample. The vast majority of uveal melanomas do not have a high mutation burden. And this contrasts markedly with cutaneous melanoma, which is one of the highest mutation rates of any cancer type. However, on the left, the red boxed area shows a few tumors with relatively high tumor mutation burden. Interestingly, this group included all of the tumors originating in the iris, so that's denoted by the black bars here, as well as the 2 cases in this entire cohort that carried germline mutations in a gene called MBD4, and those are signified here by these red bars. Now, if you look down at panel C, this color coding of the bars for each tumor indicates the relative contribution of various mutation signatures. The mutation signatures are indicators of the mutagenic agents or molecular processes underlying the development of each tumor. And if we focus initially on the 2 tumors with the MBD4 mutations, you'll see the signatures are predominantly a dark green bar, which represents a particular type of chemical change called spontaneous deamination to one of the nucleotides, cytosine, in DNA. This occurs because of the lack of function of the glycosylase enzyme that's normally encoded by MBD4. Now, if we look at the other samples in the red box here, we see a variety of pink shaded bars. Now, these represent changes to DNA caused by ultraviolet radiation. This data shows conclusively for the first time that sunlight is indeed associated with uveal melanomas occurring at the front of the eye, on the iris, but it is not at all responsible for uveal melanomas occurring at the back of the eye, which are the remainder of the samples. Importantly, this has therapeutic implications, as I will discuss shortly. So looking back to the MBD4 germline mutations, it was first shown 2 years ago by a group of Marc-Henri Stern that a uveal melanoma patient with a germline MBD4 mutation had a complete response to single-agent immunotherapy. The authors of that study speculated that this was due to the abnormally high tumor mutation burden in that patient's tumor, which was hypothesized to create lots of novel neoepitopes, that is antigens, that could be recognized by the patient's immune system, which would then kill the tumor cells. We were told of a uveal melanoma patient in Brisbane who had a remarkably good response to combination immunotherapy, and so we analyzed her DNA to see if she too had a germline MBD4 mutation. And we're pleasantly surprised to find that she did have one. And once again, assessment of her tumor showed that she had a very high tumor mutation burden, thus strengthening the association, which we know very well from cutaneous melanoma, between high tumor mutation burden and favorable response to immunotherapy. The implications of which point to a subset of uveal melanoma patients, i.e. those either with an MBD4 mutation or those with iris tumors, since they all also have high tumor mutation burdens. These subsets of patients likely having good outcomes from treatment with immunotherapy. So looking back at individual genes that are currently mutated in the uveal melanomas rather than in the germline, we confirmed that the known oncogenes and prognosis-related genes observed by others are the most commonly mutated genes. But we also found statistical evidence for the first time of 3 other genes that show mutations at higher frequencies than expected by chance. These are TP53, a very well-known cancer gene that shows a high proportion of mutations in a diverse range of tumors, as well as RPL5 and CENPE, neither of which have really been particularly well known for a role in cancer before. This figure shows the specific mutations and the relative positions of them in the latter three genes that we saw in our series of uveal melanomas. The mutations in CENPE were mostly missense changes and having as yet unknown consequences on the encoded protein. So we feel those results need to be validated in other tumors and functional work carried out to determine if they are deleterious or not. In contrast, all three mutations we observed in RPL3 were loss of function mutations, resulting in a truncated gene product. Interestingly, the work of others has shown that inactivation of RPL5 results in destabilization of p53 by allowing the oncoprotein MDM2 to degrade p53. Thus, RPL5 mutations are similar functionally to p53 mutations. And consequently, as you would expect, we find that mutations in these two genes are mutually exclusive. The therapeutic implications here are in the potential use of MDM2 inhibitors, such as nutlins, to treat uveal melanomas with either p53 or RPL5 mutations, an approach that has not been tried to date. So this figure shows the chromosome copy number changes across the entire genome for each uveal melanoma in our cohort. Pink shows chromosomal gains, whereas green indicates losses. Chromosome 3 shows the most frequent losses, and chromosome 8q shows the most frequent gains. When we put the chromosome copy number changes together with the specific gene mutations in each of the uveal melanoma tumors, we find that the samples can be subdivided into the four groups described earlier by the Cancer Genome Atlas Consortium. But we find that the assignment of tumors to a particular category is not as clear-cut or as pure as originally proposed. So, for example, some of the group 3 tumors have EIF1AX mutations. Some of the group 4 tumors have SF3B1 mutations. And we even have one group 1 tumor that has a BAP1 loss of function mutation. The two sets of curves shown here graph the relapse-free survival statistics for patients based on the molecular category of the uveal melanomas, categories 1 to 4 at the top, or whether or not the tumors simply show monosomy for chromosome 3 at the bottom. If you're looking at the top curves, categories 1 to 3 all show long relapse-free survival in the majority of patients, more than 75%, whereas 90% of the patients of category 4 will have a recurrence of the uveal melanoma somewhere in their body within 10 years of initial diagnosis. And that finding mirrors the trend we see at the bottom here, where the samples are subdivided based purely on whether the tumors carry a single copy of chromosome 3 or not. So to summarize, we find that NBD4 mutation carriers have a high tumor mutation burden. Iris melanomas also have a high tumor mutation burden, but this is due to ultraviolet radiation exposure. High TMB predicts response to immunotherapy. And so selection of patients for immunotherapy based on having either germline NBD4 mutation or an iris tumor might be a good way of stratifying patients for clinical trials or immunotherapy agents. There's also the possibility of using MDM2 inhibitors for those uveal melanomas that carry TP53 or RPL5 mutations. So with that, it just leaves me to thank the funding bodies that supported our work shown here, as well as the large team of collaborators that actually did all the work that I presented for you today. Thank you.
I want to thank Uma and Rowan for giving me the opportunity to present immunotherapy of metastatic uveal melanoma. So 50% of uveal melanoma patients develop metastases, which is mainly to the liver. BAP1 mutations, gene expression profiles, and copy number variations are associated with metastasis. Once metastatic disease appears, this lacks approved treatments. Because the tumor is often confined to the liver, different types of liver-directed treatments have been tested. Also, systemic treatments have been used and are being used, like classical chemotherapy, but targeted therapy is, of course, problematic since metastatic uveal melanoma does not have BRAF mutations, and MEK inhibitors hasn't resulted in an overall survival benefit. Immunotherapies have also been tried, and I will give you a little bit of a background to what types of immunotherapies are being used in cancer to put it all into context. So immunotherapy can of course be any type of methodology that enhances the immune activation against cancer. It can be non-specific immune stimulation via cytokines or bacteria, or it can be vaccination strategies. But the type of technique that has gained most traction recently is of course the immune checkpoint inhibitors, including CTLA-4 and PD-1 inhibitors. These have shown dramatic effects in skin melanoma, but much less so in uveal melanoma. Other types of checkpoints do exist, and currently there's trials testing those as well. And in Gothenburg, we have been working on the PEMDAC trial, which is a trial where PD-1 inhibitors and HDAC inhibitor entinostat has been combined in uveal melanoma, and we will be able to present those trial data beginning of next year, we think. But the majority of the presentation today will deal with adoptive T cell transfer, where basically immune cells are grown ex vivo and then infused back to a patient. And this work all started with us trying to develop an animal model of adoptive cell transfer that was also humanized. So we had previously made so-called patient-derived xenograft models where a tumor sample is grown in an immune-compromised mouse, and when it grows up, it's further transplanted into we have cohorts of mice which can be treated with different types of drugs, and then responses monitored. In order for us to do a mouse model of adoptive T cell transfer, we made use of the knowledge gained from many years back, where you can grow a tumor ex vivo in the presence of IL-2, and then the T cells will grow and kill off the tumor cells. And you can even get very high numbers that can be infused back to the patients, and this can give high response rate in melanoma and even complete and durable responses. But it's a labor-intensive and expensive methodology, and it can be also associated with considerable toxicities. But we anyway thought that maybe this could be modeled in an immune-humanized PDX models by basically growing the tumors first in mice, and in parallel making T cell cultures which, when tumors grew in mice, could be infused back, and then responses could be used to monitor the efficacy of the particular T cells. And this type of model we would call PDX version 2, and we could then see if this model would correlate with adoptive T cell transfer patient data, if we could find markers for response, and if we could use it for modeling combination therapies between targeted therapy or epigenetic therapy and other types of therapy, and adoptive T cell transfer. As well as it would be interesting to genetically manipulate T cells to enhance effects of those T cells that don't work in the PDX version 2 model to later be translated into the clinic. So this is just a description of the paper that we have already published, which basically shows that it is possible to obtain complete responses in PDX models using adoptive T cell transfer. But the prerequisite for this methodology is that you're using a particular transgenic animal which express human IL-2, so that the T cells can survive longer in the animal model. And when we use this model, actually this mouse model can distinguish between a responder and a non-responder to ACT. But what we didn't know is whether or not the PDX version 2 model also can be predictive of other types of immunotherapy, and if we can achieve additional effects by combining with other therapies. So we have done this recent paper published earlier this year, where we basically show that the PDX version 2 model can also be predictive of PD-1 therapies, and you can gain additional effect by combining with targeted therapy and ACT in skin melanoma. But of course, this model has primarily been used in skin melanoma. And what about uveal melanoma? To do that, we needed biopsies, and we recently published this year the molecular profiling of metastatic uveal melanoma biopsies that we obtained from the SCANDIUM trial, which is a randomized multicenter phase 3 study, which evaluates if perfusion treatment can increase overall survival in comparison with best alternative care in patients with isolated liver metastasis from uveal melanoma. This is an open surgical procedure that the surgeon does, and therefore they have been able to give us fresh biopsies that we have profiled the genetics of, and we have tried to generate PDX models as well as grown out tumor-infiltrating lymphocytes. And when we then analyze these tumor-infiltrating lymphocytes, we see that there's great inter-patient heterogeneity in uveal melanoma, we developed PDX models from 2 of these TIL cultures corresponding patients. And we could see in this case that the tumor grew even though we transfused the TILs into the mice. But in this case, the TILs could actually eradicate the uveal melanoma in this PDX version, so it's suggesting that some patients' T cells are actually good enough for adoptive T cell transfer. And this is then question, what do these T cells recognize, since it's known that the tumor mutational burden in uveal melanoma is among the lowest of all cancers. So we checked to see whether or not these T cells instead could recognize common melanoma antigens, and it turns out that many of them actually could, although it's not the majority of the TILs in the culture that they recognize these particular MARCH1 or GP100. And in a trial published 2017, also this group were able to show that there is reactivity even in patients of adoptive T cell transfers, where 7 of the 21 patients in this trial actually had an objective response. And we are currently setting up an own TIL trial in Gothenburg, where we're using an automated bioprocessor for generating the clinical grade TILs. And then the actual therapy will be via hepatic arterial infusion to get the TILs directly into the liver metastasis. And we hope to be able to start this trial around February or March of next year. So we probably will not see responses in all patients, just like the Kumula-Rosenberg groups didn't do. So we're already thinking of how we could reactivate immunity against those uveal melanoma that does not respond to adoptive T cell transfer. And the current working hypothesis is that we could perhaps use so-called CAR T cells. And what are those? Well, they are actually T cells are genetically generated to mimic signaling from the T cell receptor. So if a tumor loses the ability to show mutated antigens or melanoma-associated antigens, you will have a second chance to kill the tumor using an antibody moiety here on a chimeric antigen receptor that can recognize something on the cell surface. And then when it binds there will be a signaling domain mimicking that of the T cell receptor. And to identify tumor antigens, we mine the gene expression of both the TCGA as well as our own tumors to look for proteins expressed on the surface of tumor cells, and that were also already known CAR T targets. And what we saw in this publication is that surprisingly, HER2 is actually expressed at a quite demonstrable level in both skin melanoma and uveal melanoma. Probably has been missed because the gene is not amplified, but the protein is overexpressed. So we showed in this paper that we are able to use HER2 CAR T cells to eradicate uveal melanoma in vitro as well as in this animal model. So we're trying to now use Clinimax to generate clinical grade CAR T cells. We also, in this project, doing a slightly different variation. Here's just showing data that you could actually eradicate also uveal melanoma using CAR T cells against HER2. So we're doing a slightly different approach to the generation of the CAR T cells, and the reason for this is that there's many challenges for CAR T cells in solid cancers, including tumor heterogeneity, where the expression of, for instance, HER2 or CD19 can be lost, and then the CAR T cells can't bind to the tumor cells anymore. It's very difficult to get the T cells into the tumor. And once they are in, there is of course a lot of factors in the microenvironment that can inhibit T cell function, as well as the expansion is quite difficult in the solid tumor field. So we think that at least these two, the impaired T cell trafficking and tumor heterogeneity, can be addressed by introducing the CAR construct into TILs instead of blood-derived T cells, which is the type of T cells that are normally used. Because then the CAR TILs, as we call them, will be able to bind to the tumor cells via the CAR, but if the CAR target is not expressed, then the TCR could potentially bind. And as well, it's known that TILs can actually home into the tumors and they can traffic into there and expand in there. So this is a new thing that we're trying. And here's just an example where we have expressed the HER2 CAR on a TIL from an uveal melanoma. And when we do this, we can actually see that these TILs now become more efficacious become activated when they meet tumor cells, and they actually kill these tumor cells much more efficiently than ordinary TILs. So we're then trying to push this towards the clinic and to address the concept of toxicity on CAR TILs. We're currently using companion dogs in a first time in dog trial, FIDO trial, because dogs also develop melanoma, not uveal melanoma, but very often mucosal melanoma. That's also BRAF wild type. And currently the FIDO trial has not revealed major side effects. So we hope that we will be able to go forward with the filing of the regulatory documents for the Swedish EMA to, after the high TIL trial, start a high CAR TIL trial. So thank you so much for your attention. I just also want to thank, of course, a number of people, including the patients and their families. So Lars Ny, Lisa Nilsson, and Roger Olofsson Bagge are key members in the translational melanoma group that we had in Sweden. Roger now has his own group, but Lars continues to work together with the GMP lab and Samuel, Rebecca, and Elin here to push both TIL therapy and CAR TIL therapy towards the clinic in Gothenburg. And I also want to thank, of course, technical support and also funding from various sources, including more recently the Kirkbride Melanoma Foundation here in Perth that has supported our move to Perth. And I thank you very much for the attention.
Good day. My name is Ellen Gray, and I'm a researcher at the School of Medical and Health Sciences at Edith Cowan University in Western Australia. And I will be talking today about my research in uveal melanoma. And I would like to thank the organizers of this event for inviting me to share my work. as possibly previous speakers have introduced, uveal melanoma is the second most common subtype of melanoma, affecting the uvea layer of the eyes and usually subdivided in different types, affecting the choroid, the ciliary body, or the iris. Despite improvement on the treatment of the primary disease in the eye, the survival of uveal melanoma has not improved, with around 50% of patients developing metastatic disease, primarily to the liver, in 95% of cases. And once liver metastasis develop, survival is relatively limited to months and perhaps a year. And this is due to the lack of effective treatment of metastatic disease. In particular, in Australia, the incidence of uveal melanomas, as evident by the result of a recent analysis that we carry out, hasn't. The incidence of uveal melanoma is elevated in males with a 9.1 per million cases compared to females at 7.7 per million cases, and the age standardized rate it maintains relatively similar over the years. However, there was a significant increase over from the 80s to the 90 to 95, and then a reduction observed since in both males and females. The incidence of uveal melanoma across different states within Western Australia haven't differ significantly. However, there is a trend of that reduced incidence of uveal melanoma at high latitudes, with Tasmanian displaying the lowest incidence rate at 6.7 per million case individuals. On the other hand, survival has been maintained steady over a number of years. It hasn't been improved despite improvement on the treatment of the primary disease, with staying at an average of 80.7% of across the last 30 years. When we compare the survival from uveal melanoma across different states, we notice that Victoria displays significantly better survival of uveal melanoma, with Western Australia displaying the worst survival, and possibly this is in the back of previous studies showing that that the survival rate of uveal melanoma in rural Australia are worse due to the inaccessibility or the less access and distance required to travel to access early detection, treatment, and as well as surveillance of the disease. And this is a critical issue in Australia, and to add to the number of critical gaps on the uveal melanoma field, in particular across these different critical unmet needs in uveal melanoma that Richard Carvajal have highlighted in one of his talks. My work mainly revolves around the analysis of risk stratification and the use of surveillance strategies for early detection of metastatic disease. It is become evident that uveal melanoma can be classified in different subclasses or classes that have distinct risk of developing metastases, which ultimately would compromise the life of patients with uveal melanoma. And patients with uveal melanoma that have monosomy 3, BAP mutations, and gain in 8q have a low survival probability compared to patients with class 1 tumors. However, to define this very effective prediction tool, you need to sample the tumor in the eye, which involves very invasive procedures and is commonly not performed for various reasons, including the fear of complication from this procedure. But in addition to that, once a risk is defined, there is no, at least in Australia, there is no a consensus of the screening modality to monitor the development of metastatic disease once the treatment of the primary ocular melanoma is completed. And in this context, we think that the liquid biopsy will provide, will fill up this gap, will provide a tool in which improve the management of uveal melanoma. So the analysis of blood biomarkers such as tumor cells that you can find in the bloodstream or tumor-derived DNA that you can find in plasma of patients allows us to obtain information about the tumor in a way that through an accessible sample that can also be repeated and used to monitor regularly the evolution of the disease on a patient. Circulating tumor cells, which are these tumor derived from the patient's tumor cells derived from the patient's tumor can have been shown to carry some copy number alterations that represents the same alterations that you can find in a tumor, therefore can serve as a surrogate to or an alternative to a tumor metastasis. And the fact that we don't observe these alterations in blood cells explains also the idea that we can use the specificity of this test to define these copy number alterations that provide a risk stratification in these patients. On the other hand, circulating tumor DNA, which is pieces of DNA derived from the tumor as these cells break down, are found in the bloodstream. However, patients with primary disease have very little of this tumor DNA. Patients with metastatic disease have elevated amount of circulating tumor DNA. And from previous work from the Institute Curie in France, it was demonstrated that the amount of circulating tumor DNA in patients correlate with the tumor volume in patients with metastatic disease. In longitudinal studies that we have carried out, we can observe that circulating tumor DNA can serve as an early evidence that development of metastatic disease, in particular in this patient, a few copies of circulating tumor DNA were detectable when still no evidence of metastasis through a PET scan. However, later on, when a singular metastasis was detected in the liver, there was a further elevation of circulating tumor DNA, which was reduced once the tumor was resected. It was not it didn't disappear, and in fact, it became elevated once consistent with the detection of further metastatic disease in the liver of this patient. Similarly, we have observed in a number of patients, this is another example of a patient with, again, an elevation of circulating tumor DNA in the blood of this patient with no evidence of disease. And in fact, further elevation of circulating tumor DNA corresponds with the detection of 2 metastatic deposits. In this patient, again, this is another patient with no increase in circulating tumor DNA or detection of circulating tumor DNA consistent with progressive disease. Again, we didn't have samples prior to the development of metastasis, but we know that once it was treated with a combination of ipilimumab and nivolumab, the circulating tumor DNA reduced rapidly. However, it was not noted that there was a response to treatment in this patient, and we didn't have further blood samples, but patients in general eventually develop further disease progression. But this decrease in circulating tumor DNA after treatment with ipilimumab, nivolumab combination immune checkpoint inhibitors was of interest to us because we also observed in another 2 patients this decrease in circulating tumor DNA once they got treated with this combination immunotherapy, which is something we didn't observe in patients treated with monotherapy with pembrolizumab, which is just the anti-PD-1 inhibitor alone. And this kind of perhaps is an interesting finding that there might be a signal of certain level of control of the tumor with this combination of nivolumab, perhaps not sustained and therefore easily escaped by uveal melanoma, and therefore developing. There is no evidence of this treatment to be effective, but it provides some further interest on explore this treatment in combination with others, and I think it's in general we suggest that the application of liquid biopsies for uveal melanoma may differ depending on the disease stages, with circulating tumor cells providing information about the risk of developing metastases, but they providing information of copy number alterations and therefore serving for prognostication, especially in patients where they're doing a biopsy to the tumor in the eye. It might be slightly complicated, or the patients refuse to have a biopsy, and then the blood provides an alternative sample. But this definitely need to be confirmed in prospective studies. In addition, circulating tumor DNA provide a path in which we can monitor patients for early detection of metastatic disease, and therefore enabling either the implementation of early adjuvant therapies or early resection of singular metastases, but definitely is provides a path for early management of the disease before it becomes broadly disseminated. On the other hand, monitoring circulating tumor DNA through the treatment process may provide first signals that there might be some control of tumor, especially in a context of clinical trials and early phases clinical trial to move forward with certain drugs that may enhance the response once it's used in patients with a smaller volume of disease. I just wanted to finalize with providing an example of a patient that we also monitor circulating tumor DNA through the disease with a reduction in circulating tumor DNA once the tumor in the primary tumor was effectively treated with non-detectable circulating tumor DNA over a number of months, and with an elevation of circulating tumor DNA consistent with the development of a singular lesion in the liver, which was resected. Circulating tumor DNA became undetectable. Again, there was an elevation which coincided not with liver metastasis, but instead with a regrowth in the primary tumor, which resulted in enucleation of the eye, and then again undetectable ctDNA, which became elevated, again coinciding with later on development of 2 metastatic deposits, which were resected. However, remaining this metastatic disease was observed in the liver, and eventually the patient was treated with ipilimumab nivolumab with only a single dose due to complications of hepatic toxicity and further development of metastatic disease, and ultimately this patient passed away. But we had the opportunity to analyze the 2 tumors obtained from this patient, and important to remark that prior to that patient, this patient didn't receive any systemic treatment. However, if you we observe that these two tumors have a distinct profiles. First, one of them have a much larger immune infiltrate of T cells than the other tumor. But also when we look at these two tumors, first by inferred copy number variation, we observe the lack of monosomy 3 in the tumor from segment 2, where the tumor from segment 3 had a clear monosomy 3 in addition to a number of copy number alterations, which was clear also in whole genome sequence, well as CGH array. But a SNP array of this the segment 2 tumor clearly identified that there was an isodosomy of chromosome 3, so which evolved from this a primary with monosomy 3 as well as this other tumor in segment 3, and this one resulted in a doubling of the chromosome 3 unique copy. But this tumor in segment 2 also has unique characteristics because it was relatively homogeneous compared to the tumor in segment 3, which have high heterogeneity across the different states. And when we compare these two tumors, we also find that there is distinct transcriptional programs with tumor 3 having active oxidative phosphorylation as well as MYC target. And in fact, many of the markers associated with a class 2 tumors were active in this tumor in segment 3, while in segment 2 was less of a classical class 2 tumor, and it has a reduction in oxidative phosphorylation, preferencing the use of fatty acid metabolism. And in fact, this segment 2 tumor had a high immune related activity in their tumor cells, while the tumor in segment 3, which has a high active oxidative phosphorylation, has a low immune signatures associated with the tumor. And these results, although preliminary, and we're still trying to find more details, highlights the level of heterogeneity that the disease have even at these stages, which also pose challenges for the treatment, effective treatment of the disease. With that, I just want to finalize my talk by acknowledging the people that have done this work. PhD student Aaron Beasley carried out most of the liquid biopsy work in collaboration with research assistants and another postdoc in my lab, as well as I would like to thank Dr. Tim Isaac, which is the main ocular oncologist recruiting these patients, as well as pathologists and oncologists that have supported my work on uveal melanoma. And I also would like to thank Dr. Wei Taoling, which is a postdoc in my lab that have carried out the single-cell RNA sequencing in collaboration with Professor Alistair Forrest from the Harry Perkins Institute. I would like to thank the patients and volunteers that have got involved in these studies and have donated blood samples regularly so we can carry out this monitoring of the disease. And of course, I also would like to thank all my funders of these studies. Thank you very much.